Thank you very much for the introduction, and thank you, Dr. Cohen, Dr. Carvalho, and Dr. Lemieux for giving me the opportunity to talk about some of our most recent work, I should say, in machine learning, but in the realm of protein research. So before demystifying all the technical words that we have in this title, let's just talk a little bit about why do we care about protein research. The first thing that we know is that our bodies are composed of a vast number of different types of cells. These cells play different roles in terms of the organs that they are part of, in terms of the function of these organs. And in the end, these cells, when one of the most important building blocks of their functionality, are encoded into proteins. Proteins are these molecules that are actually originating from the DNA. So the DNA is the genetic background that you have in all of your cells. The DNA contains genes, and these genes ends up being sometimes transcribed into RNAs, which are then later translated into these molecules that we call proteins. And you can imagine that proteins are basically involved in most, if not all, of the processes that take place in the cell. They take place in terms of proliferation, division, growth of the cells, but also processing nutrients, such as glucose, for instance. So it's involved in metabolism and driving the energy of the cell. So they're a very important part of the functionality of human cells. And so proteins, a little bit like DNA, is made of these molecules, these building blocks molecules that are called amino acids. These amino acids, there's 20 in human. These 20 amino acids have different names, obviously, but also different masses, which is going to be extremely useful later on for how we actually identify proteins when we process a sample. So in the end, this brings up to one of the key words that we had in the title, which is called proteomics. So proteomics is actually this large-scale study of the proteins that we would have in any type of biological sample. And why do we want to study proteomics? Well, because it gives us a lot of information about the biological samples that we are analyzing. One of the main things that we do in proteomics is that we basically try to characterize the proteome, or the set of proteins that are part of a biological sample. And the idea behind that is that it gives us an idea of the functionality of that sample. So what is actually taking place? So we could be looking, let's say, at a tumor biopsy and want to study the proteins that are present, and gives us some insights about the disease and the special type of cancer that is taking place in that biopsy. We might be interested in simply characterizing a cell line, because we're using that as a model to study a disease such as an ALS disease, as we heard in the previous talk. But we could also analyze much more complex samples. I did mention that humans were composed of multiple cells. Well, we're composed of human cells, but we're also composed of a lot of bacterial cells. Actually, more bacterial cells than human cells, if you are curious about that. And so we could actually look into what is called the microbiome, so the set of bacterial cells that are, let's say, in your gut or on your skin, and try to look at the proteins that are present in these bacteria to try to understand what's their role and what's their interaction with the humans, us, their host. And so that's one thing, characterizing the proteome. Well, another thing that we can do, which is actually quite useful, is quantify proteins. And so one thing that we often do in proteomics is we'll have 2, 3 different set of experimental conditions. Let's say a cancer condition, so a disease condition, and a normal or a healthy condition. And we'll be interested in knowing if not only are there certain proteins that are present, but are their level different in between the two conditions. So is a protein, for instance, more abundant in the cancer sample than it is in the healthy sample, or vice versa? And this obviously gives us a lot of insights into potentially the mechanisms that are driving the diseases, but also linking it to potential therapeutic targets. Or it could be also a biomarker, something that can help us diagnose the disease that is present. And so proteomics will help us quantify the proteins and see if there are any differences between the two conditions. One last thing I want to talk about, and the third main application in proteomics, is to look at protein-protein interactions. In fact, when we look at protein-protein interactions, we are actually looking at the potential contacts that we have between two proteins that are in a given cell or a given sample. And that is actually quite useful in terms of determining the functionality of proteins, because usually when you are performing a certain function, well, proteins rarely act alone. They usually group together and they will act and form complexes of proteins that are all interacting together in terms of, let's say, processing glucose in the cell. And so we'll have these proteins that will be interacting together, and we can study them, and we can infer potentially functionality with groups of proteins doing the same function. And so what all of these things have in common is one important theme, is that the basics of one proteomics experiment is that you more or less always have to identify the proteins that are present. Before you quantify them, before you find their interaction, you have to know what is present in your sample. And there is one major widely used pipeline that's called bottom-up proteomics that is often used to identify proteins in a sample. And this is done through an instrument called a mass spectrometer. And I'm going to go briefly through this pipeline, but the way that this works is that you'll have your protein mixture, which can come again from a tumor biopsy, a cell culture, any type of biological sample you can imagine, where we extract the proteins, and then we will actually break these proteins apart into smaller molecules that are called peptides. So we break the proteins apart simply because the molecules are too large to be analyzed in a mass spectrometer. It's too difficult to analyze those, and so we need to break them into smaller pieces. Then the next step is to actually separate the peptides in terms of not having to send all the peptides at the same time into the mass spectrometer, because it would be hard to identify every single one of them at the same time. So we separate them using a biochemical technique. And like this, the mass spectrometer sees only a few peptides at a time and can actually start analyzing the ones that are present. And comes the actual mass spectrometry aspect of the pipeline, where the peptides are broken into their building blocks, the smaller pieces that I mentioned about, the amino acids, and their masses are recorded. And that builds what is called a mass spectrum here, that basically represents a fingerprint of a peptide or a piece of a protein. And so once we have these mass spectrum, we can use computational approaches to actually map these mass spectrum to a sequence of amino acid, which represent a piece of a protein, for instance. And so I'm going to spend a little bit more time on the computational methods that are used. One of the most commonly used approach to identify peptides from mass spectrum is called a database search. And there's obviously all sorts of tools that are thereafter to statistically assess the quality of a peptide or a protein identification. So let's spend more time on the database search. So database search is a concept that's been in proteomics for now almost 25 years. So it's a very old concept, but it's still the most used concept for identifying peptides. And the idea is actually pretty simple, because when you're analyzing, let's say, a human sample, well, we know that most of the human genome, and so we know most of the genes that are present in the human genome, and therefore we can anticipate the vast majority of the putative proteins that would be present in a human sample. And that's true for most model organisms, whether it's E. coli, mice, even dog. We have a pretty good idea of what is the potential proteins that would be present in our sample. And so the idea is pretty simple. Let's just try to build what would be the theoretical set of spectra that we would see if we were to analyze all of the different proteins that are present in the human proteome. So we build this gigantic theoretical database of mass spectrum that are representing the different possible peptides, because we know how they get broken into in the mass spectrometer. And then we simply try to match them with the data or the mass spectrum that we've actually acquired during our experiment. So we've acquired a given mass spectrum. We don't know what the peptide or the protein sequence associated with is. And so we'll just try to match it to theoretical spectrum from peptides that we expect, because we know we are analyzing, let's say, a human sample. And so we try to match those. And what happens is that when we align them, we're going to try to compute some sort of matching score or confidence score. The one that is the most often used is the cross-correlation score. And what happens is that we'll see here, well, it's a pretty poor match, so it obtains a low score, so we'll move on, and we'll try to match it against other theoretical spectra from our database, and the next one should be coming. There you go. And we'll have here, let's say, a much better match between the theoretical and the acquired spectrum. And what happens here is we'll get a high cross-correlation score, and we will actually the database search algorithm will actually report the peptide associated to the theoretical spectra as the one that represents the spectra that was acquired. Now, there's one issue with that technique is that the vast majority of database search approaches will basically always report one hit, the top one. So it'll report the top hit in your database search and will say, this is the most likely peptide represented in the spectra that you've acquired. But obviously, not all of the peptide spectrum matches are actually good. Some of them are actually quite poor, but it might still be the top hit, the best match of the data that you've acquired. And so what happened, the reason why this happens is many folds. Actually, I've enumerated a couple of reasons here, but there's a lot more. One of the reasons, maybe the most common, is that the peptide might be of very low abundance. So there's a certain amount of sensitivity for the instrument. So if a peptide is of very low abundance, then the signal-to-noise ratio of the data that you acquire will be poor, and therefore it will be very hard to match it against a theoretical spectrum. Another issue, probably the second most common, is the fact that once we always assume that we are analyzing one peptide at a time, but we might be at certain time during the mass spectrometry analysis analyzing two different peptides at the same time. And so the data that you acquire is not for one peptide, but actually two peptides. And so you get what is called a mixed spectrum or a chimeric spectrum, where the result will be of two sequences that have been broken into different pieces. So when we try to actually correspond this acquired mixed spectrum to the theoretical ones, well, we'll be partially matching one theoretical spectrum, partially matching another, both fairly poorly, and therefore the scores won't be that great. And so we're reporting the top hit will be fairly bad, and it might just be 1/2 of what we're actually seeing. Finally, another aspect is that I talked about proteins and that are made of amino acids, but there are also molecules that gets added into proteins and confers them some functionality. Those are called post-transitional modifications. And these molecules have a certain mass, and they change the mass spectra that are acquired. And if you don't anticipate those when you're building your theoretical database, then you will have discrepancies and will be difficult to identify those. There's obviously a lot more reasons why it is difficult to match all peptides, but those are probably some of the top ones. And so one thing that you need to do, because you're always reporting these top hits, is to try to get an estimate of your false positives in your top hits. And so one approach that was proposed around 15 years ago, and that is also still used, is to build what is called a target decoy database search. And so you have your target hits, which are the ones I was showing before with your theoretical database, but you're going to add in your database search a set of decoy hits. The decoy hits are basically fake spectra, that you will add into your database. So the decoy ones will be hits that you don't expect in your sample. So peptides, spectra that represent peptides that are simply not in human. And so one thing that you can do is simply, if you have the theoretical spectra for the peptide here, well, you can create a decoy one by simply reversing the sequence of amino acids and create the spectra corresponding to the theoretical spectra corresponding to that sequence. In fact, there are very few peptides or proteins that are palindromic in human. And so therefore, by creating these reverse peptides, then you create theoretical spectra that should not be matched when you're performing a database search. These spectra should not be present in your sample. And so once you perform a database search, once you have a hit against one of those decoy spectra, that means that you will actually have a false positive. And that gives you an estimate of your false positive rate or your false discovery rate. And so we enter the machine learning world once we have an idea of what are our false positives and what are potentially our correct hits. And there's been a lot of tools that were published in the early 2000s that use the target decoy database search strategy coupled with a machine learning tool in trying to figure out, basically, based on the confidence scores from the database search, what are the bad hits from the decoy hits and the potentially correct hits that are a subset of the target hits that we have, the matches to our theoretical database, and try to figure out if a hit, given the confidence scores it received from the database search, was actually a correct peptide identification. And the top one is here, Percolator, is probably the state-of-the-art tool that is still today the most used. Now, well, these tools are old. Yes, that's true. And that's a common thing that they share. Another thing that they share is that, well, they still have sensitivity issue. In fact, when you run a proteomics experiment, only 25% of the mass spectra that you acquire are actually matched to a peptide or a protein identification. That means that for every dollar you spend on your proteomics experiment, you basically get only 25 cents of biological information. In a world where grants are not easy to get by and money is definitely hard to get in medical or biomedical sciences, this is not an equation people like. It's a lot of money that is invested and not a whole lot of return in terms of the information. And so there's obviously all sorts of issues that could explain this, and the ones I've exposed before are definitely part of it. But there's also, and I'm not here to blame the mass spectrometry or the proteomics people. I think there's definitely a lot of effort that can be made from the computational side in terms of better mining the data that is acquired in order to yield a more sensitive protein identification. And so the tools that I've talked about that are trying to classify what are the correct from the incorrect identification share another thing in common, besides being old again, is that they actually work in silo. They work in complete isolation. So when you are analyzing a data set, the tool is given the data set, it trains on it, it tests on it, and it reports the result. It doesn't use any other data set that was acquired. It doesn't use any other external information. So it basically just works one data set at a time. And that's a major issue because there's a lot of information out there that can be useful in determining whether a peptide or a protein identification is actually present in your sample. In fact, one of the reasons for that is probably the years at which these tools were published. Sharing proteomics data back then was not really a thing. There were no public repositories that were available, and if there were very few data sets that were there. And so access to shared resources was not really in the mentality back in then. It is much more today. And in fact, one thing that can be quite useful in terms of information that we could bring in to try to assess more accurately the confidence of a peptide or a protein identification is probably protein interactions. There's a lot of information that is encoded in protein interactions. And so we have an example here of, let's say, the ribosomal complex. So this is a large set of proteins that are color-coded in this figure that assemble together in order to create more proteins. So they form this complex, and you have representations of different protein complexes here in the graph, or a directed graph on the right side. And the idea is pretty simple. If in my sample, I see a lot of proteins that are part of the ribosome, well, if I have identifications that are also related to ribosome that are kind of borderline, I'm not sure if those are good or not, then I should be pushing myself and pushing the confidence that the proteins that are borderline are also present. If the ribosomal proteins are there, then I should see the proteins that I'm interested in. And so the tool that we came up with is called MS Proteini, and the idea behind that tool was actually to integrate protein interaction information in order to increase the sensitivity of protein identification in proteomics. And this is work that was performed by a fantastic master's student in my group, Francesca Barry, who will be graduating soon and will be looking for jobs. So if you're hiring, then you can write down her name. I'm sure she'll be happy to entertain any interviews. So basically, the hypothesis behind our work is that the confidence of a given protein identification should increase if in your analysis you keep seeing proteins that are known to interact with the protein you're trying to assess. And the analogy for this is, let's say you're going to a conference. This is the closing cocktail of the conference. You've just saw the last keynote talk. It was fantastic, but you know the keynote has a flight to take and is going to leave soon, so you don't have a whole lot of time, and you want to talk to that person about their work because you are dying to collaborate with them. And so that person here is Karen. But you're not too sure if this is Karen because it's the first time you saw Karen. You were at the end. It was a large conference. You were at the end of the room. It wasn't very easy to see what Karen looked like. And so the room is packed. Karen is putatively talking with a bunch of people, and you don't have the time to engage into a conversation with a group of people that you don't know. So you're really aiming for Karen. And so you look around and you go like, well, I don't know, she's turning her back. I don't know if it's her. Should I investigate deeper? And what you actually see is that the two people Karen is talking to are two of its known collaborators that you know very well. And so that should increase your confidence that the person you are looking at right now is indeed Karen. And therefore, you can approach her with full confidence and jump in their conversation and assure yourself that indeed the person here is Karen. So obviously, in that example, Karen is the protein that we're trying to identify. So we're not too sure if the protein that is present in our sample, we're not too sure if the protein that we have in our sample, the identification is correct. But if we look around in our sample and we see known interactors of that protein, then that should boost our confidence that the protein that we were looking at is indeed a correct identification. And so our goal was actually to design an algorithm that would incorporate these protein interaction information with mass spectrometry data and confidence scores that come from database search in order to improve the sensitivity of the mass spectrometry analysis. And so basic pipeline of MS Proteini is as follows. You basically get a protein sample. It's analyzed through mass spectrometry. We perform a typical database search. We use Comet as the tool here. And then to these results from the database search, we will integrate protein-protein interaction information, in this context from the BioGrid public repository, which is one of the biggest repositories for protein-protein interactions. We also integrate the database search results, the protein interaction information, into a supervised learning framework. In this case, it was a support vector machine that will basically output in the end the matches that are of high confidence in terms of the peptide and protein identifications. So for our tool, in terms of benchmarking it and analyzing the data that we had on hand, we use the HEC293 dataset, which is a type of cell line that is very commonly used in mass spectrometry to benchmark different techniques and approaches, simply because we have a very good idea of the proteome of that cell line. So one of the first things we did, and I think that what most people should do when they're trying to build a machine learning algorithm with data sets like this, is to look at how the features were actually playing a role in the classifications. And so one thing that we first did was simply to look at two of the main features for the classifier, the cross-correlation score, so the quality of the match between a peptide, theoretical spectra and an acquired spectra, and also simply the number of proteins that are known to interact with a given protein that we're investigating that were present in our sample. And what we can see is that, well, as expected, obviously, so in red you have the decoy hits, and those are situated towards the left side of the plot where you have very low cross-correlation scores. So cross-correlation scores that show poor matches because those are basically fake spectra and they shouldn't be matched. Higher ones will have a lot of blue hits, which are deemed target hits, so potentially good identifications. If we move a little bit closer here, we see that there's a region between 1 and 2 cross-correlation scores that if we move on the y-axis, which is the number of proteins that are known to interact, we'll actually have, I guess the colors here are not easy to contrast, but there's a lot of space here where there are actually no red matches or no decoy hits. And so that actually shows that by using the number of interactions, we can potentially discriminate more accurately what is a correct or what is an incorrect identification. In fact, very rarely in any proteomic studies will people report identifications with cross-correlation scores below 2. So that was something that was actually helping us thinking that there was room for progress in that specific area of the data. And so we built our support vector machine, a very simple one, with 3 features: the number of interactions that were present in the sample, the cross-correlation score, and something called the delta correlation, which basically represents the difference between the score of the top hit of the database search and the second best hit. The idea is pretty simple. If you have a very good top hit, then it should be way higher than the second, third, fourth hit. And so if that score is high, that difference is high, then you believe that you have a good hit. And so once we decided on our features, we obviously needed to decide on the training of that classifier, and that's actually pretty straightforward for the negative examples because we have all these decoy hits that represents negative identifications. But the positive training example, it's not that trivial, in fact. For the positive training examples, well, we don't really have any because we are trying to figure out if the matches that we have are indeed correct or not. And so we kind of have to build a set from a proxy that would be enriched for positive identifications. And so what we did is we built a set based on peptides that were very abundant. If you look at peptides that are very abundant, those tend in general to yield more correct identifications from incorrect ones. And so the main idea behind that is that we would have here a positive training set that would be enriched for positive examples, but not necessarily pure of those. And so based on these, we've actually decided to benchmark our tool against the state-of-the-art tool that is currently available for assessing protein identifications, which is Percolator. And what we see is that not only were we actually outperforming Percolator at any false discovery rates that are deemed reasonable. Typically, proteomics data sets that are published will never be reported with a false discovery rate higher than 1%. And so we can see that all of these false discovery rates, we were actually outperforming, identifying more spectra than Percolator. But I guess even more interestingly, and that was the point that we were trying to make, is that there is a lot of information in the protein interaction, because if we remove protein interaction as a feature, we see that we have a huge loss of performances in between our two tools. Now, if we move on to, in terms of number of unique different peptide sequences that we were identifying, we see that we still have an increase over Percolator, and that again, the number of protein interaction was actually playing a role in the performances of the tool. And finally, if we go to the protein level, we see that we've overall, because the peptides are the small pieces of the proteins, we see that at the protein level, we actually outperform Percolator again, and then once again, protein interaction was playing a role in our ability to classify good from bad hits. Again, across the entire range of high confidence false discovery rates. Now, obviously, identifying more things, having a higher sensitivity, it's all great, it's all amazing, but we wanted to know a little bit more about where the gains were and the biases that we were including in our analysis. In fact, by including protein interactions into our analysis, there was a high risk that we were biasing our analysis against proteins that have no known interactions. There are proteins that are still not very well characterized and will have no interactions in the database like BioGrid. And so we were curious to see, would we simply completely toss those out? Would we completely bias our data set that we would only be able to identify proteins that have known interactions, which would not be ideal, obviously. And so what we looked at is, first of all, determining the proteins that had no known interactions in BioGrid, and we had about 3.5% of the data set that we were studying that had no interactions in BioGrid. And we see that at very low false discovery rates, so at close to zero false discovery rates, we actually identify about half of those 3.5% proteins. So we're actually quite happy about that. We do see that there is most likely some bias in terms of proteins that we're identifying, because as the false discovery rate goes up, we see more and more proteins that don't have any interaction. So we might have included some bias in terms of proteins that have no interactions that are maybe biased against, but it's not because they have no interactions that are simply tossed out. So we still are able to confidently identify them, even if we don't have any interaction information about them. Now, the other thing that we were really interested in seeing, where were our gains against Percolator? What kind of Percolator hits were we rescuing that they were rejecting? And so what we looked is the distribution of confidence scores from Percolator. So that's the full set of scores that were outputted by Percolator. And you see here where the purple distribution actually starts, which might not be easy to see with the lights, but this line right here is the line where these scores were rejected by Percolator. They were deemed non-confident. And so what we're interested to see is, among those, what was the proportion that was rescued by our tool, by MS Proteini? And what you can see here, and it's going to be a little bit more easier in the next slide, is that the vast majority of the hits that were rescued were actually towards the highest bad or rejected scores from percolator. So if we actually look at that specific region, and we look at the proportion of hits that were rescued by MS Proteini amongst those that were rejected by percolator, we see that at the highest rejected scores, we were actually pretty close to 1. So we were saving the vast majority of the hits that were deemed borderline by percolator. And as we move down, we have these hits that are less and less confident, and what we can see is that our proportion goes down. And so we're not just simply uniformly sampling across the hits that percolator is rejecting, but we're biased, or we have more rescues amongst the hits that were borderline. So again, it's hinting that the protein-protein interaction information is maybe coming in and basically giving the little boost that you needed in terms of the confidence assessment of the identification. So the very last result I want to share, I don't want to separate you from the pizza too long, but the very last result I want to share is related to how much information did we have about the proteins that we were identifying. So it's one thing to identify more proteins, but did we get more information about the proteins that we were looking at? Because in the vast majority of the time, in proteomics, you simply identify a few peptides that correspond, so a few pieces of the proteins that you're interested in. You don't get the entire protein sequence, the entire set of amino acids. And so we were curious, are we making more identifications across the whole scale of proteins, or are we actually seeing improvement in the number of peptides or the fraction of peptides that we are seeing for each protein? And what we can see is that across a wide range of false discovery rates, we actually saw a higher sequence coverage or a higher number of amino acids that were perceived in the experiment than percolator. So again, that gives us more information about the identification. It is actually pretty useful when you're looking at the things like post-translational modification. So sites on proteins where other molecules are added. So that gives you a higher power to detect those sites and basically gives you more biological insights about the sample that you're looking at. So in summary, well, I hope I've been able to convince you that, first of all, machine learning approaches are actually quite useful in terms of assessing the confidence of peptide and protein identification in mass spectrometry and proteomics, but that most importantly, that we need to be a little bit more clever about how we analyze this data and integrate more information to it. And obviously, protein-protein interaction is a good example of the type of information that can help us assess better what is a good from a bad protein identification. But it's not the only thing. Obviously, protein interaction is one thing, but we could be looking at other pieces of relationships between genes or proteins. We could be looking at co-expression of proteins, for instance. So if we know that at a given time in the cell, two proteins are known to coexist, and at a given time they don't, well, if you see and you identify one of the two proteins that are known to coexist, well, that should give you more insights that the other protein would also be present because they're known to be co-expressed. We could also be looking at things like protein co-occurrences. So proteins that are known to be in the same cellular compartment, or even at another level, proteins that are known to show up in the same experiments over and over across the globe using different instruments. So you can mine data sets that are not publicly available and say, well, when they use all these sorts of instruments, when they see a protein, they see the other one, well, that's information that we should consider in terms of our protein identification. And obviously, one thing that we are interested in is also to look at less characterized protein interaction networks or interactomes for species, let's say like bacterial species, where we don't have a whole lot of information. How would our algorithm perform? So do we need a lot of information, a large interactome that's very comprehensive, or can we study samples where we don't have a whole lot of information about the relationship of the proteins? So with this, I would obviously like to thank my lab and Francesca Barry, who did all of this work. Also our collaborator for this study from the Figues lab, Jibin Ning, who processed the mass spectrometry and proteomic sample and supplied them to us. And the funding for this project was from NSERC and Compute Canada supplied the computational time. And one thing to conclude, we are currently recruiting in my group. So if you're interested in applying machine learning techniques to biomedical or bioinformatics context, then we're definitely recruiting, and not only for our curling club, actually, but for our actual research lab. We're not that great at curling, but if you are, then definitely join in. Well, with this, I'd like to thank you very much, and I'll be happy to answer any questions. Thank you.